So my name is Michael Rogawski. I'm a professor at the University of California, Davis School of Medicine. And in past years at this symposium, I have spoken about pharmacological thoughts for the treatment of SLC6A1. But in the last several years that Amber has been running these wonderful meetings, we've made a lot of progress as a community, and we have a number of interesting therapeutic strategies that are in development. And I thought that this would be a good time for us to step back a second and say, what happens once we've discovered a potential new treatment? What do we do with that treatment to make it available to patients? And it just so happens that at UC Davis, I teach a class in drug discovery and development to graduate students, chemistry majors and biology majors. And I thought I'd give you an abbreviated 101 version of that course very quickly here during my talk. So that'll be the first part of what I'm going to speak about. And then in the second half, I'm going to give an example of a case where an academic project developed into a drug that ultimately was FDA approved. And there are really two lessons from that story. The first one is a somewhat pessimistic lesson that I hope we can overcome as a community, and that is that it takes a heck of a long time to go from the idea to the actual therapy that patients can pick up at the drugstore. But the optimistic side of that is that you can do it. It is possible. And a new treatment can come into existence that never existed before. And that's really a part of the endowment of mankind. Once that treatment does become available, it really can benefit those patients for which the drug was discovered originally, but it can have a lot of other ramifications and utilities in other areas. So that's the outline of what I'm going to speak about. And in this slide, I try to break up the process of drug discovery and development into two major sets of activities. The first is the discovery phase, and that's what you've been hearing a lot about at this symposium today and in past years. We've been talking about what are the strategies that we might think about to treat SLC6A1. And the types of studies that are done in this phase of the discovery phase for new therapeutics are of the kind that are done in research laboratories in universities and research institutes. They're based on experiments that are run by the individual scientists running the laboratories and based upon the culture of science that we learn when we are in graduate school getting our PhDs. But once we have the therapeutic in hand, there's a whole other set of what we refer to as development activities that, as academics, we don't really learn too much about. It's the purview of industry. But it's just as important because for the drug to get to patients, we have to go through this regulated development process. And the rules and regulations that relate to development have evolved over the years. I calculated out the amount of time that these regulations have been in existence, and I was surprised to find that it's been 116 years that we've had the drug regulatory regime in the United States, starting from the earliest days of the FDA with the Food, Drug, and Cosmetics Act in 1906. And over the years, there have been changes that Congress has imposed into the regulatory regime. Something very happened 60 years ago with the introduction of the requirement to show that your drug actually works in patients. Prior to that time, all you had to do was show that either it was not adulterated, that is, it didn't have any impurities in it, or that it was safe and not going to cause harm to your patient. But 60 years ago, with the Kefauver Amendment to the Food, Drug, and Cosmetic Act, we now had a regulatory requirement that the sponsor, that is the entity that is developing the new drug, had to demonstrate that the drug actually worked to treat the disease. And of course, that changed everything and made it a lot more difficult to develop a drug. But at the same time, it gave patients the confidence that when they were receiving a drug, it had been studied and shown to be efficacious. So this regulated part of the drug development pathway has a lot of nuances to it. And it consists of two major parts. There's laboratory work that has to be done. And that work is done in a somewhat different way than what we do in the laboratory when we're in the discovery phase. We have to follow what are called GLP requirements, or good laboratory practice requirements, where we have to keep exquisitely detailed records and the ability to have the work audited by the regulatory authorities. And it makes it very difficult to do those kinds of studies. And similarly, when we move ultimately into patient studies, we have to carry out the work according to what are referred to as good clinical practice requirements. So all of the things that are done in the context of the clinical trial have to follow regulations that are defined by the federal government. And I'd be remiss to say that we also have to actually manufacture our product according to regulated requirements, which are called GMP requirements, or good manufacturing requirements. Okay, so this is a schematic illustration of the way a drug originates in the imagination of a scientist, and then moves through a process of non-clinical development. That is development in the laboratory before any patient has been exposed to the product, and then ultimately moved into clinical development, that is into studies in patients, and then to the marketplace where patients can purchase the drug in their pharmacy. And this slide is perhaps the pessimistic part of my talk. And this shows the timeline over which this typically occurs. And you can see that the total timeline, and this can vary enormously from one product to the next. But on average, it's perhaps 20 years of time. So that's a very long period of time. Where if you say that the identification of the target on which to develop the drug and then find the drug and optimize the drug might take 5 or 6 or more years. But then once we move into this regulated phase, this can take typically 10 years to occur. Now, as I said, I'm hoping that we as a community can figure out ways to do this more rapidly, and maybe there are some ways to do that. But in my experience, it's a very, very long haul. Okay, so what are the stages that occur after discovery? Well, first of all, we have to come up with a lead molecule, and this may involve chemists working together with biologists to optimize the chemical structure. And then we move into testing in animals. The testing that occurs in animals is fundamentally to understand how the body handles the drug, whether the drug can be absorbed and get into the bloodstream and then ultimately into the brain for a CNS drug, like a drug to treat a developmental epileptic encephalopathy like SLC6A1. And we also have to show that the compound is safe. We don't want to expose any patients to the drug before we have some evidence that there aren't going to be serious toxicological issues. And all of that data, and it's a considerable amount of data, is then packaged up into a document. It used to be a very, very thick binder. Now it's all electronic. And we submit that to the FDA in the form of what's known as an IND, which stands for Investigational New Drug Application. And the FDA sends it to their team of experts, clinicians, toxicologists, pharmacokineticists, and so forth. And they look it over and make sure that the product has some rationale for treating the disease, but most importantly, they're protecting patients and making sure that the product is going to be safe for that first patient that's going to be exposed. And that's a very big deal when you take a drug that's only been exposed to animals and then try to bring that into a patient. And there have been some very serious situations where patients responded differently from the animals and got into serious trouble. So all of this is codified by law, federal law called the Code of Federal Regulations. You can go up to the internet and look at the various sections. It's very complicated and difficult to navigate. But these are the general types of data that we include in the IND application. First of all, we have to explain what we're planning on doing to investigate the drug, how we're going to study it. We include what's called an investigator's brochure, which is giving all the information that we know about that drug so that a physician who is prescribing that drug or using that drug in the context of the clinical trial has all the information available to make sure that if there is an untoward event of some kind, that they can figure out the best way to handle it based upon the information that's available. We have to include a protocol for the clinical trial on how we're going to study the new drug. The FDA's clinicians are going to go over that, and they're going to assess that clinical study to see whether they feel that the data that is going to be generated from that clinical study are going to be sufficient to give them the information they need to whether to approve the drug or not. So that's very important. Then we have a very big section in this IND document called CMC, Chemistry, Manufacturing, and Controls. And we have to have very detailed information about how we manufactured the drug substance, which is or what's called the API, active pharmaceutical ingredient, and also the drug product, which is the formulation, the pill or injection or what have you. We really have to tell the FDA where we got all of the starting materials for the manufacturing, what we did to manufacture the drug product, and then we have to give data on the purity and the stability of the compound. We want to make sure that it doesn't break down before it's given to the patients in the clinical trials. We have then all of the pharmacology data and toxicology data that I told you in animals. And with this set of data, then the FDA can take it. The FDA actually only gets 30 days to decide whether to allow you to go into the clinical trial or not. If they are unhappy with the set of data, they'll put you on clinical hold, and then you have to provide additional data before they allow you to dose that first patient. So with that as a background, I'm going to give an example of a case where we actually went through all this process from the point of identification of the molecular target to the discovery of a molecule that actually acts on that molecular target in the brain. We didn't stop there. We then, with a set of data in animal models that indicated that this product could be used to treat epilepsy, we put together the pieces that allowed that molecule to go through the regulatory stages and ultimately become an FDA-approved drug. So this story actually starts in Bethesda, Maryland at the NIH, where I was a research scientist before I moved to UC Davis. We had some ideas about a class of molecules called neuroactive steroids, and we thought, okay, these molecules potentially could be used to treat epilepsy. For those of you who know about the field of neurosteroids, you know that these act on our old friend, the GABA-A receptor that you've heard so much about today and at other SLC6A1 meetings. And we have so many experts on the GABA-A receptors here at this meeting. And so this is a drug that binds to the GABA-A receptor and can increase its activity, increase inhibitory activity, can potentially dampen seizure activity. And we conducted studies like what you heard about before the break with electrophysiology to prove that the neurosteroids did, in fact, potentiate GABA-A receptors. We looked at a wide variety of different animal screening models that we used to determine whether a drug could be effective in the treatment of epilepsy. And this type of drug, in fact, was very potent at doing this in a broad range of animal models. And so all of this began in 1986. And we had a group of molecules that we studied and were interested in. There was another molecule that was being developed by a group of scientists in California at the time. They conducted an early clinical trial showing that there could be activity. It wasn't clear what was going on in that clinical trial. But in 2004, we actually founded a spin-off company from our work. By this time, I was ending my research career at the NIH, and I was moving out to California to UC Davis. And with a group of scientists at Yale University, we put together a proposal for a company that was ultimately funded by venture capital. And this is, I think, an important lesson here. And that is that you can do a lot in academia. And there are many, many different ways in which an academic can obtain funding for the early discovery phases. You heard about some of those from Vicki Whittemore this morning. But at some point, you have to move into a company environment. It's just not feasible to do the kind of regulated studies that are required in an academic context, although you can take it, as I say, quite far. In any case, the venture capital money was used for a variety of different clinical trials. And then things got very difficult because while the early clinical trials showed activity, one of the clinical trials didn't reach statistical significance. And at that point, we were really hanging on a knife edge, and the whole thing could have gone for naught. The venture capitalists had put millions and millions of dollars, and we probably, under normal circumstances, would have had to close up shop and go home. But our venture capitalists were persistent, and they kept providing funding to the company. Ultimately, the company was able to go public on the stock exchange and raise additional monies. Then they decided to change from developing an epilepsy drug for the broad range of epilepsies and try to develop a drug for the rare epileptic encephalopathy called CDKL5, developmental and epileptic encephalopathy. And this is a condition that, like SLC6A1, is a rare disorder. It is associated not only with epilepsy, but with intellectual disability, with a variety of behavioral disorders. Seizures can develop early on in life, sometimes just a few hours after birth. In other cases, in the first few months of life, in a variety of seizure types. Tonic seizures can have epileptic spasms, generalized tonic-clonic convulsions, and focal seizures. So a wide variety of different seizure types. And of course, there was no medicine to treat these patients. All the medicines weren't working. It was largely a refractory epilepsy condition. But it was fortunate that the drug was able to be tried in these individuals. And the first study was just a very, very small open-label study with no placebo group in 6 individuals with the disorder. And you can see the results there. On the left, you're plotting the reduction in seizure frequency, and you can see that one of the patients actually got worse in the study. A couple patients didn't seem to show much effect, but there were a number that did, and that encouraged the development of a second study that was a controlled clinical trial, and it was referred to as the global phase 3 clinical trial. Now, this is very different from the situation that would have been required to develop a drug for epilepsy generally. In that case, you'd have to have thousands of patients be exposed. And you'd have to have at least 2 at what are called adequate and well-controlled trials. That's the language that the FDA has in their law. And that's a very high bar. But the FDA recognizes that for rare diseases, like the ones we're talking about here, it's just not feasible to find thousands of patients. It's not feasible even to do 2 clinical trials. You're lucky if you can do 1 clinical trial with a small number of individuals. And that's the optimistic message that I wanted to say, that the regulators understand this, and they're willing to work with communities like this group to figure out ways to get drugs to help our patients. In any case, the clinical trial that was done enrolled only 101 patients, just a fraction of the number that would be required in an ordinary epilepsy study. And fortunately, the trial had a very robust and positive outcome. The test drug, ganaxolone, caused a 30% reduction in seizure frequency, and the placebo had a very small 4% reduction in seizure frequency. So it was just this one study in the 100 patients. The caregiver impressions, as you can see here, indicated were all over the map, but skewed toward more improvement in the ganaxolone group, which was statistically significant at the p equal to 0.015 level. Now, another lesson that you can take from this study is that the patients were drawn from all over the world. In order to get the patients sufficient, even for this 100-patient study, required going to Europe, going to Russia, you can see there, Poland, England, France, Australia, and so forth. So it was truly a global study that was required here. And this is the result. In March of this year, the FDA put out this announcement on their website that ganaxolone, it goes by the brand name of Ztalmy, was approved, and it received what's referred to as orphan drug designation, which is a big stimulus to companies to undertake these kind of difficult projects with rare diseases. They are able to sell their product exclusively for 7 years. That's the biggest incentive, and there are other ones as well. Also, in this project, the sponsor received what's referred to as priority review. This was a breakthrough compound because it was the only drug that would treat this rare epileptic encephalopathy, and so they gave it an accelerated 6 months review. Normally, it would take them 10 months to review the product. So it's a long road. It takes a long time, but it is possible. So with that, I think I will stop. And are we going to have questions at the discussion? Okay, perfect.